Today we discuss about the introduction to food safety and the microorganism that spoil the food or comes under the food microbiology. So today we discuss about the introduction to food safety and microbiology. So first of all, what are the food safety or food hazard? Actually, it is divided into a causative categories called a hazard. The organism microbes which spoil the food, right? Which spoil the food, or some microbes which are helpful in the food industry, helpful for the food industry, that also comes under the food microbiology. And there are microbes which cause or spoil the food, that comes under a hazard. So there are two types of microbes. There are two types of microbes. First microbes, there are two types of microbes. First one which help in the food production, right? And second one which spoil the food, right? Spoil the food, or that comes under a hazard. These all are the two types of microbes. Clear? So biological hazards are bacteria, molds, naturally occurring toxin. These are biological hazard. And chemical hazard are chemical like petroleum, herbicide, pesticide, heavy metals. These are chemical hazard. And physical hazards are glass, rocks, wood, splinters. This comes under physical hazards. So in this video we mainly discussed about the biological hazards that happened by the microbes. In 1919 versus 1997, the hazards caused by the microbes are tuberculosis, pneumonia, diarrhea, heart disease, liver disease, injuries, stroke, cancer, bronchitis, diphtheria. So the ratio of the disease in 1919 to 1997 is more as compared to 1999. In 1997 it is more. In the 10 leading causes of death in the United States in 1900 and 1997. Clear? So microorganisms such as bacteria, parasites, virus, yeast and mold, these all are a microorganism that seen via a naked eye. Clear? This is the chart of microbes. These are the atom molecules, virus, bacteria and archaeal organisms, unicellular algae, colonial algae, protozoa, fungi, multicellular organisms. These are microbes. Actually, where these microbes are found, they are present everywhere in our environment. Everywhere. We can't say that here there is no microbes. In our environment, everywhere there is a microbe. It's either soil and water, it is utensil, equipment, gastrointestinal tract, food handlers. In everywhere having a microbes. Sources of fecal contamination of foods. This is the main sources of the fecal contamination of foods, because there are a house fly, right? House fly, mosquitoes, these insects or flies, what they do, they sit everywhere. It is that fecal is present there or food is present there, they sit everywhere, and contamination occur by these insects. So what happened? Meat animal, processing plants, production, product contamination, food ingredient. What happened? From poultry, dairy cows, humans, sewage, everywhere, from everywhere where this food contamination, product contamination will be occur. Microorganisms in food. Microorganisms are important in many different ways. They are important, already I told you, microorganisms are important and harmful. Both are present in a food industry. So how they are important? How they are important? Pathogenic or disease causing microorganisms can cause illness. These are the important. No, this is not important. This is the actually for microbes they are important. In terms of microbes, this pathogenic, their activity are pathogenic. So for microbes it is important. They spoilage microorganisms cause of food to smell, taste and look unacceptable. Fermentation microorganisms produce desired food product. So pathogenic and spoilage, these are the harmful effect of the microbes, whereas fermentation is a useful effect of the microbes. Other microbes do nothing in foods. So these are three different ways how they are important and how they are harmful. So pathogenic and spoilage, these are a harmful effect, and this fermentation is the useful effect of the microbes. Clear? Next is controlling growth. How the microbes are controlled. The microbes for food nutrient content for growth, microorganisms require the following. They also require a substance for their growth, for their metabolic activity, because without the metabolic activity there are no organisms who are survived. Either they are microbes or prokaryotes or eukaryotes, they all having a metabolic activities for their division, for their survival. Clear? And for the metabolic activity, they require this material like water, source of energy, source of nitrogen, vitamin and related growth factors and minerals. Understood what they require? They require water, source of energy, source of nitrogen, vitamins and related growth factors, mineral. Now food activity. Optimal pH for the growth of microbe is 6 to 8. What? The optimal growth for the growth of microbes is 6 to 8, and for the disease causing bacteria it is 4.6 to 9.5, and for the spoilage bacteria it is 1.5 to 9.5. These are the activity of the microbes according to their pH. Now pH growth range in foods. Moles having survive at the range of 11. Yeast is from 1.5 to 9.5. Lactic acid bacteria, they are active at a 3 to 12. Staphylococcus is from 6 to 10.5. E. coli is in acidic medium and basic medium, both they can be survive. But Vibrio species survive at a neutral, approximately survive at the neutral pH. The mild heat required since spores are inhibited by acid. High heat required in order to destroy spores. Controlling growth. With the help of temperature, we can control the growth of the microbes with the help of nutrient of food, we can control the growth of the microbes, and according to pH also we can control the growth of microbes in the food industry. The optimal growth of the microbes, the temperature is 37 degrees Celsius. But there are some thermophilous bacteria are also produced. Microbes are also present that grow in hot condition. Mesophils like warm condition around body temperature. Psychrotrophs can grow at refrigeration temperature, and most pathogen are mesophils. Temperature classification based on optimum temperature for growth. Optimum temperature for growth: psychro cold, meso means middle, thermo means warm, tropic means growing, diuric means withstand, phil or philic referred to love, means they want to grow at low temperature, they want to grow at high temperature. Clear? And these are the graph at which the bacteria will grow. This is the temperature, and this is the hyperthermophil which grow at high temperature. This is a thermophil which grow at high temperature but not that too much high. And mesophil grow at normal temperature, and psychrophil at a low temperature. Clear? Controlling growth: temperature lower than 41 degrees Celsius. Temperature that is lower than 41 degrees Celsius, we can control the growth. Bacteria ceases to multiply but do not die. Freezing can cause cell wall damage. Freezing can cause cell wall to damage. Temperature higher than 135 degrees Fahrenheit, the bacteria will be die if heated for a sufficient time. And if the temperature will be increased at the 135 degree Fahrenheit, the increased destruction with longer times and with high temperature. Now time. Under optimal conditions, some bacteria can double every 20 minutes. The colder the storage temperature, the longer the potential shelf life. Potentially hazardous food should not remain in the danger zone for more than 4 hours during the entire food handling process. These are the diagram of how the bacteria will be divided in every 20 minutes at the formula of 2 to the power n, because every time they double from the initial step. Clear? Now generation time under optimal condition. At 37 degrees Celsius, Bacillus species, the generation time of Bacillus species is 28 minutes. And for E. coli, the generation time will be 12.5. And for Staphylococcus species, it is 27 to 30. Clear? And this is the growth phase of the bacteria. This is lag phase, this is log phase, and this is stationary, and this is the death phase. How the bacteria will be growth and how they will be die. In lag phase, they require nutrient, they start dividing but not too much. In log phase, they divide and increase their number. In a stationary phase, they form a static condition, and in death phase there is depletion of the nutrients, so they are die. Oxygen is one of the another important factor for the microbes for the growth, and how we can control the growth of microbes with the help of oxygen also, because aerobic bacteria need oxygen. If we don't supply the oxygen, then aerobic bacteria will not grow at that in that environment. Anaerobic bacteria go only if oxygen is absent, and facultative will be at both. So tolerance to oxygen in the surrounding environment, according to tolerance, there are 3 types of bacteria. Next is water activity. Moisture in a food system that is available for microbial growth and chemical reaction, the relative humidity of a food. Clear? This all are a controlling growth water activity of common food. What? Water activity of common food. Now, what are the controlling factor from which we can control the activity of the microbes? That is a FATTOM. That is, F is food, A, right? F is food, A is acidity, T is temperature, and next T is time, O is oxygen, and M is moisture. So these are the term by which we can control the growth of the microbes in food industry. Clear? Now, intervention, combined effect. Predictive microbiology, the study of interactive effect of factor affecting microbial growth. Single factor is also affect the microbial growth, and there is a if we want the effect of two or more factor at a time, or the interactive effect, then this effect is more effective as compared to single. So additive effect, there is synergetic effect and antagonistic effect. There are three types of effect in food industry. Food industry interventions. Ingredients have good specification and control of incoming ingredients. What? Ingredient having good specification and there is control of incoming ingredient. They having the process of an adequate thermal process to destroy microorganisms so that they not spoil the food. Have an adequate packaging system to protect your product. After processing, when processing will be completed, have control of distribution, distribution and a system for tracking and recalling. Provide necessary information for consumers. What they provide? They provide necessary information for consumer. And farm to farm food safety and quality program. Ingredient control. How we control the ingredient? Specification for ingredient makeup. We consider the quality, physical and microbial contamination level of the ingredient, how the ingredient are affected, which microbes affect which ingredient, we have to know. And during the processing, we have to control the effect of the temperature, air, according to that, and control the activity of the microbes. So, a specification of ingredient makeup, quality, physical and microbial contamination level. Clear? Thermal processing control. In thermal processing control, we control the cooking, pasteurization, commercial sterility process, retort system, aseptic processing and hot filling. Package integrity. In package integrity, measurements are made to ensure the package is of food sanitary quality and can maintain a hermetic seal, such as visual inspections and torque measurement for your container. During the packaging, there is much necessary to see the hermetic seal. Clear? To see the hermetic seal. And during the thermal process, there is necessary the pasteurization, commercial sterility. Clear? Thermal processing control. In thermal processing control, low acid food should have a process to eliminate millions spores of Clostridium botulinum. Example, most meat, vegetables and dairy product. Process often at 230 degree Fahrenheit or above. Means there is a high temperature. It is a thermal processing control. How we control the microbes here? With the help of thermal. The bacteria which grow at low acid food. And in acidified food, not require too much a process to eliminate Clostridium botulinum. Example, pickled products, mayonnaise, for acidified food, pickled product mayonnaise are required. Process often 180 to 205 degree Fahrenheit. Pasteurization, hot filling. Last one is high acid food. For high acid food, they not required to have a heat process. Example, sliced orange, condiments. Process often this all occur at a high temperature, so that bacteria will be spoiled at this temperature. For low acid food, the temperature that are required is 230 degree Fahrenheit or above. For acidified food, the temperature will be at 180 to 200. And for high acid food, it is 180 to 205 degree Fahrenheit. Now pathogen. General role of microbes that present in a food microbiology. There is a commensal, ubiquitous, harmless or beneficial. Already I told you the microbes are of two types. They are harmful or beneficial. In spoilage, the food in spoilage what happened? They cause or harm the food. The bacteria, microbes which cause the harm of food. So cause food to become inedible due to change in color, flavor, odor, appearance or texture. Grow to high levels and break down food component. What happened here? In spoilage, the bacteria grow to high levels and they break down food components for their own utilization. Commensal organisms that have reached a high population. Different products have different spoilage. Different products have different spoilage flora. Not single organism can only cause a spoilage of the food. There are different types of microbes. They cause a spoilage of food according to the type of food or according to the temperature that are present in environment. So the beneficial effect of the microbes are used as an aid in producing desirable characteristics in food, such as fermentation. Fermentation is a useful term or useful food that are break down or produced by the microbes. Like for example, bread, idli, dosa that are fermented with the help of the yeast, right? So these are a useful product. Vinegar. Pathogens cause foodborne illness. These are a harmful effect. Pathogen cause foodborne illness. Microorganisms that are usually associated with the presence of pathogens are called indicators. Most E. coli do not cause illness. Food safety control hazardous to the consumer. What food safety cause hazardous to the consumer? A foodborne hazard is biological, chemical or physical properties that may cause a food to be unsafe for human consumption. Food quality control deterioration of food to attain an unacceptable state. Food safety control hazards to the consumer. A foodborne hazard is a biological, chemical or physical property that may cause a food to be unsafe for human consumption. Food quality control deterioration of food to an unacceptable state. Foodborne illness. Foodborne illness is in the United States is associated with 46 million illness in a year and 3,25,000 hospitalization in a year, 3,000 death in a year. This all data is from the foodborne illness. Common foodborne pathogen are in bacteria. The pathogen are E. coli, Salmonella species, Staphylococcus, Shigella. These are bacteria. And virus, norovirus, rotavirus, hepatitis C. Parasites, Giardia, Cyclospora, Cryptosporidium. 80 to 90% of foodborne illness from bacteria come from just 4 bacteria, that is Campylobacter, Salmonella, Clostridium perfringens and Staphylococcus aureus. These are the main 80 to 90% of the foodborne illness factor that responsible for the foodborne disease. Illness mechanism. First, infection. Microorganisms are ingested and then cause illness. Intoxication. Toxins are produced by the pathogen usually in the food. When food is consumed, illness occurs. Even if microorganisms are killed, toxin can still remain the food. So that the food eaten by the people, the toxin go inside the body and they cause a illness. And that illness is known as foodborne illness. Bacteria, already I told, the bacteria that are cause foodborne illness are E. coli, Salmonella, Listeria, Campylobacter. Clear? And these are the bacteria, how much cases and death? E. coli cause hemorrhagic choleritis. Cause infection, incubation period 2 to 4 days, symptom diarrhea, HUS, TPP, and contaminants are milk, meat, fruits, vegetable, water. Clear? Salmonella cause 10 foodborne illness, incubation period 6 to 48 hours. What? 6 to 48 hours. Symptom, nausea, fever, diarrhea, arthritis, contamination, milk, meat. Next is Listeria monocytogenes. Same here also. The symptom is different. Here vomiting is also comes in the symptom. Vomiting, diarrhea, meningitis, septicemia, miscarriage. Contaminants, vegetables, milk, cheese, meat, seafood. From this all we see that meat and milk are a most contaminant food material that contaminated by the microbes. Clear? So when we eat these two items, we clear that we heat them before we eat or drink this at high temperature, so that the microbes present in the milk and meat will be destroyed. Next is Staphylococcus aureus. Cause intoxication, incubation period 1 to 6 hours, symptom nausea, fever, diarrhea. Another is Clostridium botulinum. They cause foodborne disease. Prevention of foodborne illness. Cook the food. Cook all meat, poultry and egg to at least 160 Fahrenheit or high temperature. Other than spore forming bacteria or all bacteria or microbes are killed quite easily with the heating at 160 Fahrenheit. Avoid cross contamination. Do not cross contaminate one food with another. Keep raw food totally separated from cooked food. Chilled food. Keep foods cold. After cooking, chill foods as rapidly as possible. Remember that cooking has destroyed most of the bacteria, but spore forms that are resistant to cooking may become very active and can proliferate rapidly. Cleaning. Wash fruit and vegetables and all food possible. Personal hygiene. People working with food should wash their hands regularly. Wear hairnet, plastic gloves, etc. Spoilage organism, already I told you, bacteria, fungus, protozoa are a spoilage organism. So this all are about the food microbes that are present in food. Okay, hope this all are clear. So today we stop here.